you are welcomed in biology class, and our today's topic is function of the tubules. Now in the previous class we studied about the structure of kidney, that a kidney consists of millions of nephrons, and if we talk about the structure of nephrons, then it has Bowman's capsule, glomerulus, and tubules are there. Now, then we talked about the structure of the tubule, and that proximal convoluted tubule and distal convoluted tubule, and Hanley's loop is there. So now in this class we will study about the structure and functions of all these structures present: PCT, DCT, then Hanley's loop. As you can see in the diagram also, this is the structure of a nephron, and here you are able to see that there is a distal convoluted tubule and proximal convoluted tubule. As you are able to see in the diagram, this is proximal convoluted tubule which is going downwards. Then there is distal convoluted tubule, and then there is Hanley's loop which is forming a U-shaped structure. And this Hanley's loop, it has two limbs. One limb is known as descending limb which is going downward, and the other limb is known as ascending limb. And there is another structure known as vasa recta, as you are able to see here. So this is the structure of a particular or a typical nephron. So proximal convoluted tubule, which is also known as PCT, and if we talk about the types of cells present, so it has simple cuboidal cells, and these cuboidal cells have brush border. So this is a cuboidal cells, and this is the brush border we can say. So brush border is there, and their function of this brush border is to increase the surface area for reabsorption, because as the filtrate has to pass through these tubules, what is the main function of these tubules? To reabsorb, and for reabsorption there is need of more surface area, and that is possible because of the presence of simple cuboidal brush border epithelial cells. Now, when this filtrate, it will pass through the proximal convoluted tubule, nearly all the essential nutrients and 70 to 80% of the electrolytes and water, it will be reabsorbed by this segment. So maximum reabsorption is taking place in the PCT. And now this PCT, its function is to maintain the ionic balance and the pH of the body fluid. And how it will be done? Means first function is that the maximum reabsorption is taking place, and the second function is to maintain the pH and ionic balance. And how it would be done by PCT? It will secrete hydrogen ions, ammonium and potassium into the filtrate. So means these substances will enter into the filtrate, and by absorption of HCO3, means it will absorb HCO3. So there will be balance of pH and ions. Now next is Hanley's loop. Hanley's loop is the site where the minimum absorption takes place. So reabsorption is minimum in Hanley's loop, and but this region, Hanley's loop, it has great importance. Why it is of great importance? Because it maintains the osmolarity and the osmolarity of the medullary interstitial fluid. Now if we talk about the structure of kidney, so this is the portion which is known as cortex, and this portion is known as medulla. So what is maintained? Osmolarity is maintained where? In the medullary interstitial fluid. So these are the portions. Medullary interstitial fluid is present in it, and the osmolarity of it is maintained by Hanley's loop. Now this is about the ascending limb, and the Hanley's descending limb is also there. This is impermeable to water, so it will not allow the water to come out, and but it is almost impermeable to electrolytes. So what is there? Descending limb of loop of Henle is permeable to water but almost impermeable to electrolytes. So it will allow the water to move, but it will not allow the electrolytes, and in this way the filtrate, as it moves down, it will get more concentrated. Why? Because it is allowing water, so water is coming out and the filtrate is becoming more and more concentrated. Whereas ascending limb, it is impermeable to water, but it will allow the electrolytes actively and passively. And in this way, as the concentrated filtrate pass upward, it gets diluted due to the passage of electrolytes. So what we are able to see here, that from these two ascending limb and the descending limb, when this filtrate is passing, so one limb, that is descending limb, it is allowing water to permeate, whereas it is not allowing the electrolytes. And when we talk about the ascending limb, it is impermeable to water, and in this way this concentrated fluid, it is passed upwards and it gets diluted. Now next tubule, which is known as distal convoluted tubule, and its function is a reabsorption of sodium and water, and it also reabsorb HCO3, and it secretes hydrogen and potassium ions and NH3. So what we are able to see, that some of the ions are absorbed and some of the ions are secreted, and the main function is to maintain the pH as well as the osmotic balance. Now the next part, it is collecting duct, and this is the diagram which is showing a reabsorption and secretion of major substances at different parts of the nephron. This is the structure of a nephron. So this is proximal convoluted tubule and this is distal convoluted tubule, and these are the arrows which are showing that these substances, they are absorbed and these are secreted in the proximal and distal convoluted tubules. Now next turn is of collecting duct. Now collecting duct, it is a long duct and it will extend from the cortex of the kidney and it will move towards the inner part of the medulla. And what it will do, it will absorb large amount of water, and in this way concentrated urine will be produced. And this segment, it also allows small amount of urea. This small amount of urea, as we are able to see in the diagram also, here it is mentioned urea. So small amount of urea is also allowed to enter into the medullary interstitium. And why there is need of urea? Because it keeps the osmolarity and it plays important role in maintaining pH and ionic balance by selective secretion of hydrogen and potassium ions. So in the whole, what conclusion we have got from this, that the main function, when this filtrate is passing through the Hanley's loop or through the ascending or the descending limbs, some of the ions are reabsorbed and some of the ions are secreted, and in this way when this urine, it will flow through these parts, then counter current mechanism will be developed and reabsorption will be there and concentrated urine will be produced by the body. Now next we will study about the mechanism of concentration of the filtrate. Means when this filtrate is passing through the tubules and through this Hanley's loop and descending loop and vasa recta, so how it is concentrated? We know that mammals, they produce concentrated urine, and the main function of concentration is done by Hanley's loop and vasa recta. And I think it is clear to you what is Hanley's loop. It is a hairpin, U-shaped hairpin like structure, and it has one more structure that is known as vasa recta, and both these Hanley's loop and vasa recta, they play great role in producing the concentrated urine. So what will happen when this filtrate, it flows through the two limbs of the Hanley's loop? So this is Hanley's loop, one is coming downward, so it is known as descending. The other loop, it is moving upward, so it is known as ascending. So when the filtrate is passing through these two loops, it is moving in opposite direction, as we are able to see. Here the filtrate will come downward and in this loop it will move upward. So the filtrate is passing or moving in opposite directions, and in this way it is forming a counter current mechanism. Then next tube that is present, it is vasa recta. So what this vasa recta will do, Now this vasa recta, it will also help in maintaining the counter current mechanism. Means the vasa recta also and the Henle's loop both are playing great role in maintaining counter current mechanism. There is a great proximity or understanding between the Henle's loop and the vasa recta, and therefore they are able to maintain the counter current. And when the counter current is maintained, then there is increase in the osmolarity, that is towards the intermedullary interstitium. What we are able to see in the diagram also, that this osmolarity is, it is increasing from 300 ml, as you are able to see, 300 m osmolarity per liter, to it is near about 1200 m osmolarity per liter. So we are able to see that this increasing osmolarity towards the intermedullary interstitium, because this is the medulla region, because in the kidney this is the cortical region and this is the medullary region. So the osmolarity, it is increasing from 300 to 1200 m osmolarity per liter, and this gradient, it is caused by sodium chloride and urea, as it is shown in the diagram also. Sodium chloride is there and urea is there, which is responsible for maintaining osmolarity. Now this NaCl, when it is transported by the ascending limb of Henle's loop, as we are able to see in the diagram, there are two loop-like structures shown. One is of vasa recta and the other is nephron. So these two structures, they will help in maintaining osmolarity and concentrating the urine. So here this gradient, it is mainly caused by sodium chloride and urea. Sodium chloride, it will be transported by the ascending limb of Henle's loop, and in which is exchanged with the descending limb of vasa recta. So there is Henle's loop and डिसेंडिंग लिम्ब ऑफ वासा रेक्टा. एक की असेंडिंग लिम्ब है और दूसरे की डिसेंडिंग लिम्ब है, और दोनों ही क्या कर रही हैं, NaCl को ट्रांसपोर्ट कर रही हैं, एक्सचेंज कर रही हैं. सो NaCl जब रिटर्न होगा, व्हेन इट विल बी रिटर्न टू द इंटरस्टिशियम and by the ascending portion of the vasa recta. In this way, small amount of urea will also enter in the Henle's loop, and then again it will also be transported back. So in this way, there is facilitated flow of substances from the Henle's loop and vasa recta, and their function is to maintain the counter current mechanism. And why there is need of such mechanism? क्यों ये आयंस जो हैं बाहर आ रहे हैं या अंदर जा रहे हैं? टू मेंटेन द osmolarity, to maintain the concentration gradient, and such type of gradient is necessary to concentrate the urine or the filtrate. And human kidney, it can produce urine near about 4 times concentrated than the initial filtrate formed. In the previous class we studied about that near about 180 liter of filtrate is produced or filtered by the kidney, but when the urine is passed out, only it is in an average adult, it is 1.5 liters. So you can imagine the situation that how much concentration has taken place. So human kidney, it produce largely concentrated urine from, if we compare it with the initial filtrate formed. Now next topic is a regulation of kidney function. So who is controlling? We have studied that kidneys, it has nephrons, then in the nephron there are tubules, there are vasa recta, then there is Henle's loop, ascending limb, descending limb, and all these are in helping in making the urine concentrated or making the filtrate concentrated. But how the function is regulated by the kidneys? So these kidneys, they are functionally doing function or an in an efficient way, and they are regulated by the feedback mechanism of the hormones. Now I think you are familiar with the feedback mechanism. Feedback mechanism means when there is need of a particular hormone, then that is released, otherwise that is reabsorbed, and this is known as feedback mechanism. So urine production or the production of the filtrate, it is also depending upon the feedback mechanism of the kidney or the hormones, and it is regulated by the hypothalamus, which is a gland, then JGA, and to some extent heart also plays important role in regulating the kidney functions. Now, osmoreceptors are present in our body. Whenever there is change in the osmolarity, these osmoreceptors they will receive that. And how these osmoreceptors become activated? When there is any change in the blood volume or the body fluid volume and ionic concentration. Means whenever there is more amount of water in the blood or the body fluid volume or the ions are more, then what will happen? Will become active. Now, because as there is more amount of or there is less amount of water in the blood or body fluid volume, so osmoreceptors have become active. Now, excessive loss of fluid from the body, if it happens, if excessive loss of fluid is lost from the body, then what will happen? These receptors they will become activated, and in this way they will stimulate the hypothalamus. Up now it is a gland, so it will release a hormone, antidiuretic hormone known as ADH, and it may be called as vasopressin. Now this hormone it will reabsorb the water from the later parts of the tubule, because what has happened? Excessive loss of fluid has taken place. Now there is less amount of water in the blood, and water is required for many of the activities. So what should be there? That there should be no excessive secretion or loss of water from the body. And how it can happens? It is controlled by ADH, means hypothalamus. It will release a hormone known as ADH. Now this ADH it will reabsorb the water from the later parts of the tubule, and in this way there will be increase or there will be no diuresis. And if there is increase in body fluid volume, if more amount of water is present in the body or in the blood or body fluid, then what will happen? These osmoreceptors they will be switched off, means they will not be activated. And if these osmoreceptors they are not active, then ADH it will be suppressed. And this ADH, now what will happen? Now it will give the constrictory effect on blood vessels, and in this way there will be more increase in the blood pressure. And if there is increase in the blood pressure, then more glomerular blood flow, means kidney will filter the more blood, and thereby GFR, glomerular filtration rate, will become more. सो आई थिंक इट इज़ क्लियर टू यू कि वेनेवर जब भी बॉडी के अंदर फ्लूइड ज़्यादा हैं, एडीएच एक्टिव हो गया और उसने क्या किया, जो है वाटर की जो एब्जॉर्प्शन है उसे क्या कर दिया, क्योंकि लॉस हो चुका है तो एंटी डायूरेटिक हार्मोन एक्टिव हो गया और उसने जो है वाटर की रीएब्जॉर्प्शन को कहाँ से, लेटर पार्ट से वाटर को लेना शुरू कर दिया. एंड इफ देयर इज मोर फ्लूइड इन द बॉडी, ADH will get suppressed, and now the blood will come with the great pressure, and again there will be filtration. So JGA it also plays great role, because we have talked that heart, then hypothalamus and JGA, these three are involved in feedback mechanism for regulation of kidney function. So about hypothalamus we have studied. Now let us have a look on the JGA, that how JGA controls the functioning of kidney. Whenever there is fall in glomerular blood flow or GFR, so what will happen? Whenever there is decrease in glomerular blood flow, then GFR it will become activated and JG cells will release the renin. About JG cells we have studied in our previous class, that JG cells are the special cells, so they will secrete the renin, and this renin it will convert angiotensinogen, which is present in the blood, into angiotensin 1 and further to angiotensin 2. Means we are talking about the JGA. जब भी डिक्रीज हो रहा है ग्लोमेरुलर ब्लड फ्लो के अंदर, JG cells एक्टिव हो गई. Now they will release renin. This renin will convert angiotensinogen into angiotensin 1 and angiotensin 2. Now these angiotensin 1 and 2, this angiotensin 1 is converted into angiotensin 2. Now angiotensin 2 it is a vasoconstrictor. So what it will do, जो है it will increase the glomerular blood pressure and thereby GFR. blood pressure increase हुआ, मोर ब्लड आया तो ग्लोमेरुलर फिल्ट्रेशन रेट ज़्यादा हो गई। इसके अलावा एंजियोटेंसिन 2 इट विल ऑल्सो एक्टिवेट द एड्रीनल कॉर्टेक्स, एड्रीनल ग्लैंड के कॉर्टेक्स को, and that will also release aldosterone, and this aldosterone it will reabsorb sodium and water, and this will lead to an increase in the blood pressure. So this is known as a renin angiotensin mechanism. आई थिंक इट इज़ क्लियर टू यू। फॉल हुआ फिल्ट्रेशन रेट में, जेजी सेल्स एक्टिव हुई, रेनिन प्रोड्यूस हुआ, रेनिन ने एंजियोटेंसिनोजन को एंजियोटेंसिन 1 और 2 में कन्वर्ट किया, और एंजियोटेंसिन 2 वासो कॉन्स्ट्रक्टर का काम करेगा, एंड ग्लोमेरुलर ब्लड प्रेशर क्या हो जाएगा, इंक्रीज हो जाएगा। Now the third is how heart help in controlling the regulation of kidney. So whenever there is increase in blood flow to the auricle of the heart, it can cause the release of arterial natriuretic factor, means whenever there is more blood flow in the auricle or the atrium, then a factor ANF will be released. This ANF it will do the vasodilation or the dilation of blood vessels, and because of that there will be decrease in the blood pressure, and in this way this when there is decrease in the blood pressure means किडनी जो है उसकी जो फंक्शनिंग है वो क्या हो जाएगी, स्लो हो जाएगी. सो हेयर इट इज़ ऑल्सो एक्टिंग एज अ फीडबैक मैकेनिज्म. Now micturition. Now urine it is produced by the nephron. Now this urine from the nephron it will go to the collecting duct, and from the collecting duct ureters, and then it will reach to the urinary bladder, and it is stored in the urinary bladder until there is a voluntary signal given by the central nervous system, means our brain is involved in it. A voluntary signal will go to the brain, to the CNS, and now this signal it will initiate a stretch on the urinary bladder as it gets filled with urine, means when the bladder is full एक मैसेज जाता है ब्रेन के पास में, एंड नाउ दिस सिग्नल इट इज़ इनिशिएटेड बाय, किसने इनिशिएट किया, क्योंकि ब्लैडर के ऊपर स्ट्रेच या प्रेशर आ रहा है, एंड इन दैट रिस्पॉन्स जो स्ट्रेच रिसेप्टर्स ऑन द वॉल्स ऑफ द ब्लैडर दे विल सेंड सिग्नल्स टू द CNS. आई थिंक इट इज़ क्लियर टू यू कि जब ब्लैडर फुल है, ब्लैडर ने क्या किया, स्ट्रेच प्रोड्यूस हुआ, और उस स्ट्रेच ने किसके पास मैसेज कन्वे किया, CNS के पास, एंड नाउ दिस CNS it will lead to the contraction of smooth muscles of the bladder. Urinary bladder के जो smooth muscles हैं they will get contracted, and there will be relaxation of the urethral sphincter. Sphincter it is a valve, now causing the release of the urine. So there will be this removal of the urine from the body, and this process of release of urine it is called as micturition, and the neural mechanism it is called as micturition reflex, because this is under the control of CNS. This bladder it send message to the CNS. Now this CNS it will give them this response, and there will be, earlier there was stretch or contraction, and now there will be release of the muscles, relaxation of the muscles of the urethral sphincter. This sphincter it will get relaxed, and because of that this urine will be removed out of the body. So process is micturition, and the neural mechanism it is called as micturition reflex. And we have talked that an adult man it produces 1 to 1.5 liters of urine per day, and urine's composition it is light yellow in color, it is slightly acidic, and it has characteristic odor, means it has some particular smell, and urea is present which is about 25 to 30 gram is excreted out per day, and various conditions also affect the color or the concentration of the urea in the urine. And if we talk about the analysis of urine, it is helpful in many of the clinical diagnosis. Many of the disorders can be studied by diagnosing the urine, especially we can study the malfunctioning of the kidneys, means if the kidneys are working properly or not. Malfunctioning means when they are not working properly. So for example, if there is glucose in the urine, then what it shows that the patient or the person is suffering from the disease diabetes mellitus, and which is also known as sugar. there are some other organs also which helps in excretion. The kidney, it is removing urea or uric acid or ammonia depending upon the type of organisms. And now if we talk about, there are lungs also which are removing the gaseous waste from the body. Liver, it is removing the poisonous substances or the toxic substances which are produced in the body. And skin, it is removing the waste which are soluble in water in the form of sweat. So we can say these are the various other excretory products, or the other excretory products um, other than kidneys. Now if we talk about the lungs, these lungs, they remove carbon dioxide, and they also significant quantities of water every day. You might have observed that during the winters, when you exhale air on the mirror, there is some moisture comes on the mirror, and that moisture is because along with this CO2 some of the water is also removed. Then if we talk about the liver, which is the largest gland, and it secretes bile containing substances like bilirubin, then biliverdin, cholesterol, and steroid hormones, vitamins, and drugs. And these substances, they are removed from the digestive tract as waste. So liver is also removing the waste from the body. Lungs, they are removing gaseous waste along with water. And then skin, sweat, and sebaceous glands. We know that sebaceous glands are present under the skin, and these sebaceous glands, what they do, they eliminate the secretions, and these sweat which is produced by the sweat glands, and in this from the sweat glands water comes out, or the watery fluid comes out, which contains sodium chloride and some urea, lactic acid, etc. And we know that the function of the sweat is to maintain the body temperature, but along with the maintenance of the body temperature, wastes are also removed from these sweat glands. Now the sebaceous glands or the oil glands, they secrete substances like sterols, hydrocarbons, and waxes through sebum, and this secretion provides a protective oily covering for the skin. And some nitrogenous substances are also removed by the saliva. So these are the various organs other than kidneys which are playing role in excretion. And the next topic is disorders of the excretory system. All the body organs are very important, and if there is any defect in any of the organ, the body doesn't work properly. So it is necessary to have a knowledge of the proper functioning of these systems. And as we are studying about the excretory system, so there may be some disorders. For example, malfunctioning of the kidneys means when the kidney doesn't work properly, then what may happen? Urea may get accumulated in the blood, and that condition is known as uremia, which is highly harmful, and that may lead to kidney failure. And in such patients, what is done? Urea is removed by the process known as hemodialysis. How hemodialysis is done? Blood is drained from a convenient artery. Now it is pumped through a dialyzing unit. आफ्टर एडिंग, अब देखिए बॉडी से बाहर जो है ब्लड आ रहा है, एंड इट मे गेट कोएगुलेटेड, इकट्ठा हो सकता है, क्लॉट बना सकता है. सो व्हाट इज एडेड? एंटी कोएगुलेंट इज एडेड, and the name of the anticoagulant example is given here, heparin. So during the hemodialysis, when the blood is drained from a convenient artery, it is added with a substance known as anticoagulant, and now this blood, it will be pumped into the coiled cellophane tube, means thin plastic tubes are there, and these tubes are surrounded by the dialyzing fluid. So what will happen? When the blood is flowing through the coiled cellophane tube, and outside what is present? Dialyzing fluid is present, and that have the same composition as that of the plasma except nitrogenous waste. So let us have a look on this. This is a dialyzer. So these are the cellophane tubes. Now this blood is drained from the artery, and now it is flowing through this cellophane tubes, and here this is dialyzing fluid. So this fluid, it has the same composition as that of the blood which is passing through this cellophane tube, but there is only one difference, that the nitrogenous waste is present in this blood which is flowing through the cellophane tube, whereas this fluid, it is free of nitrogenous waste. So what will happen? this nitrogenous waste, because of high concentration in the blood, it will come out into this fluid, and in this way this blood is free from the nitrogenous waste. And the cleared blood, it is pumped back, because जो बॉडी से लाया गया तो कौन लेकर आई, आर्टरी से, and now through the vein it will be added back to the body. And now what will be added, एंटी हेपेरिन। पहले क्या ऐड किया, हेपेरिन, क्योंकि कोगुलेशन नहीं होनी चाहिए थी, बट बिफोर एंटरिंग इनटू द बॉडी द ब्लड इज एडेड विद एंटी हेपेरिन विच इज नेसेसरी सो दैट इट मे नॉट गेट कोएगुलेटेड इनसाइड द so that it may come to its original composition and with the original properties. क्योंकि ब्लड की ओरिजिनल प्रॉपर्टी क्या है कि जब भी ब्लड बाहर आता है तो वो क्या हो जाता है, क्लॉट बना देता है. सो एंटी हेपेरिन इज एडेड सो दैट इट कैन रीगेन इट्स ओरिजिनल प्रॉपर्टी. Now next disorder is renal failure. Sometimes this kidney transplantation is done, and why it is done, that kidneys they fail, they stop functioning, and in this way a kidney from the donor is taken, and preference is given that the donor should be a close relative. Why there is need of a close relative? Because our body can identify the foreign cells, and the cells can be destroyed by the defense mechanism of the body. So if the donor is a relative, then there are less chances of the immunity system to deactivate that organ. So the preference is given to the close relative. Now, modern clinical procedures have increased the success rate of such a complicated technique. Means new technologies have come, and because of that, organ transplantation has become very easier. Next disorder is renal calculi. In renal calculi, stones or the insoluble mass of crystallized salts are formed within the kidney. And the next is glomerulonephritis. In this, what will happen? These glomerulus, they will become inflamed, and because of that the kidney will not work properly. So these are the disorders of the excretory system. This is all about today's class. Thanks and have a nice day.